All right, well, hello everyone. Thank you so much for coming to my talk today. My name is Kiran, and I'm going to be talking about my master's thesis project, which I'm doing at Cal State University, Northridge. And my project is on evaluating the non-lethal effects of giant sea bass. So to first quickly tell you a little bit about myself, I am originally from the East Coast. I'm from a small town that you likely haven't heard of in central Massachusetts. And the reason that I just mentioned this is because, growing up, I didn't really spend that much time around the ocean, except for our annual kind of week-long trip to the beach on Cape Cod to see my grandparents. I never really thought about the ocean that much. And most of my life, until the time I was 18, looked a lot like this upper right-hand corner picture here. That was a farm that I spent a lot of time at as a kid, learning practical life skills, such as how to milk a cow and card wool. But I was really lucky to grow up in a place where we had a lot of outdoor space, and the way that I expressed my love for the outdoors as a kid growing up was through playing a lot of sports. And when I did decide to go to college, I ended up committing to play field hockey and run track and field at a small liberal arts school in central Pennsylvania. So again, no ocean yet. But the narrative of my story changed pretty quickly when about a month into my freshman year in college, I took a field hockey stick to the head, got a pretty bad concussion, and that followed me around for the first 2 years in college. To the point where after my sophomore year, I actually decided to take a year off and kind of regroup a little bit, recover from the traumatic brain injury, which actually turned out to be the best thing ever, and the moment in my life where marine biology became a real option for a career for me. So when I all of a sudden had all of this spare time on my hands, I decided that it'd be pretty cool to go to the beach for as long as I could. And with that, I actually got on the first airplane I had ever been on, flew down to a turtle conservation camp on the northern Pacific coast of Costa Rica. I stayed there for about 4 weeks and worked in a turtle conservation site, which was amazing, and I loved every moment of that. And once I got back home, I sent off some emails to a place at the Cape Eleuthera Institute in the Bahamas, and I got very lucky. They had a grad student that needed an extra hand on one of her projects. So about 3 days later, I found myself on a plane down to the Bahamas for 2 months to work on tagging and tracking bonefish around the island of Eleuthera in the Bahamas in an acoustic array. So after those experiences got me really thinking about the ocean as an ecosystem and a thing that I could study and make into a career, I decided to transfer schools to the University of Rhode Island. It's a little more of a coastal school. It actually had marine biology as a major. And I had many wonderful experiences in my 2 years as an undergrad at URI. Probably the most notable of which was a summer fellowship that I had back in 2018, where I got to travel with my advisor down to the British Virgin Islands, and we spent 3 weeks studying the behavior of these guys right here. Those are called bridled gobies. And this was the first time in my life that I really got to see the scientific process from start to finish through a project, which was very cool. And I had a lot of fun doing that, and I learned a lot. Felt like graduated college. I quickly relocated to the West Coast to Dr. Mark Steele's Fish Ecology Lab at Cal State Northridge. And based on kind of some reading and some grant applications I've written in my last 2 years of undergrad, I knew that I kind of had an interest in the non-lethal effects of predators. So basically I took those ideas and I modified them. I thought about them a little more in kelp forest context, and I came up with my master's thesis question of how does giant sea bass presence in an ecosystem impact prey behavior? And that brings us up to the present of my master's thesis. So before we start thinking about this question, we really need to think about what kind of effects a predator can have on an ecosystem. So generally, a predator can have what is known as lethal effects or non-lethal effects on a system. So lethal effects are sometimes also referred to as density-mediated predator effects or consumptive predator effects. And this occurs in situations where a predator is consuming a prey and reducing that prey's abundance. And that can be compared to non-lethal predator effects, which are sometimes referred to as trait-mediated or non-consumptive effects. And non-lethal effects occur in scenarios where predators are impacting a prey's behavior, activity, or development just by their presence in an ecosystem. So I'm going to walk us through an example of each of these. For lethal effects, I'm using a scenario from Estes and Duggan's 1995 paper. They had sea otters as a predator. Sea otters like to eat sea urchins, and sea urchins live on and around giant kelp in kelp forests, and they eat the giant kelp. So when you take out the sea otters from this system, essentially you end up with an explosion, an overabundance of sea urchins, and very little giant kelp left. So in this scenario, you have sea otters consuming their prey. They're changing the density of their prey, and that in turn is causing some underlying change in the ecosystem. On our right-hand side, we have an example of non-lethal effects. This is a scenario from Dr. Elizabeth Maiden and colleagues' 2010 paper on coral reefs. And on most coral reefs, or at least any that I've ever been to, you have lots of herbivorous fish living on the coral and kind of sheltering in the coral branches as they feed on the algae on and around the coral. And in a healthy reef system, you'll have lots of reef sharks, these apex predator figures, kind of cruising by intermittently. And because these predators are sort of an ambient threat in the system, the herbivorous fish often don't go very far from the branches of the coral to obtain algae food, even if there is algae that's available further away. However, if these predators are removed from a system through something like overfishing, all of a sudden the herbivore fish have a lot less to be afraid of, and they're willing to go a lot further away from safety to get their food. So it's that trade-off. And this is what that looks like in reality. These are two satellite images, one on the top showing a situation with sharks, and the one on the bottom showing a situation without sharks. And on the top, you can see these sort of colorful dots are coral reef patches, and the white rings around them are what are known as grazing rings. So that represents essentially the furthest that any herbivore is willing to go to obtain their food. And all of this dark area in between the patch reefs is filled in with algae. But if you move to the bottom, where there aren't sharks kind of keeping those herbivores in their place, you still have these patch reefs, but you have no grazing rings and essentially no algae in between any of the reefs because it all gets mowed down by the herbivores. So in this scenario of non-lethal effects, you have sharks' presence eliciting a behavioral response from the prey, and that in turn is affecting the underlying ecosystem. And that can be compared to the lethal effects scenario where you have a predator consuming the prey and changing the abundance or density of it, which changes the ecosystem. So it's two different mechanisms that produce similar results. And we're focusing on non-lethal effects for my master's thesis. It is important to note that lethal and non-lethal effects occur simultaneously in all scenarios, but just depending on the players in the system, essentially the pattern that you see emerging is going to take one shape or another depending on the characteristics of your players in the study system. So in my study system, I have a predator of giant sea bass. And for my prey, I'm going to be focusing on common predator fishes of California. Giant sea bass are thought to have really diverse diets. They eat a wide variety of crustaceans and fishes. They've even been noted eating things like kelp and small sharks. They're monsters. But because I think fish are more likely to show behavioral responses to predators, I'm going to be focusing on just the fish prey for my thesis. All right, so for a little bit of background about our main predator, the giant sea bass, their scientific name, Leos apex predator in the northern Pacific. They range through most of Southern California and down into Baja, Mexico, and inhabit depths of up to 40 meters deep. Giant sea bass were a really popular fishery in the 19th and 20th centuries. Because of that, they saw a major population crash around the 1930s when the fishery collapsed. And since those days, they've been functionally extinct from the Southern California ecosystem. But luckily, in more recent years, some protections have led to the beginning of their recovery. So a moratorium declared on the fishery in the 1980s and a subsequent gillnet ban in the 1990s have done good things for the populations of giant sea bass. And this is just a figure that demonstrates this from House and Colleagues' 2016 paper. This is data from the Channel Islands Research Program showing the number of giant sea bass seen on scuba surveys per year around Catalina Island between 1965 and 2011. And the general easy takeaway that I want you to see from this is that there were no giant sea bass for a really long time, and now there are a few more giant sea bass. And it's gotten to the point where you can start to see aggregations of them relatively reliably around areas of Southern California and around areas of Catalina Island, which is where I'm focusing my work. So we really don't know very much about giant sea bass and how they inhabit the kelp forest ecosystem. We barely know anything about their long-range movement, except that they can move really long distances pretty quickly. We do also know that they seem to show seasonal site fidelity to aggregations at specific locations. And from my perspective, from what I want to study, I think that these aggregations are providing temporal and spatial patches of risk to the prey that are at these sites year-round. And I think that this sort of patchy risk scenario could enhance the anti-predator responses of any of the mesopredator prey that live in these areas. So going back to my main question of how does giant sea bass presence impact prey behavior, we know that giant sea bass presence is probably having some effect on the behavior of these mesopredator prey, but I want to know to what degree that's happening and to what degree we're really seeing those anti-predator response behaviors in our prey. And to understand that, I also want to know what factors are best predicting the degree of the prey response that we're seeing. So what's going into how severe or not severe these prey fish are responding. And I have 7 factors that I'm mostly going to look at. I have them sorted into sort of predator characteristics, prey characteristics, and environmental characteristics. So I think that things like the density of giant sea bass, the proximity of a giant sea bass to a prey, and the axis that the giant sea bass is on in relation to the prey. So what I mean by that is whether the giant sea bass is along a horizontal axis to a prey or whether it's above or below the prey, are all things that could change how severe the anti-predator response we're seeing in a fish is. For the prey, I think that the size of the prey, whether or not it's bite size for a giant sea bass, and the site fidelity of a prey could impact these behaviors as well. And for the environment, I think that time of day could be a factor, and I think that the habitat complexity could also be a factor. So getting into my methods, for I'm just going to run a quick site assessment. I have 3 main goals with these site assessments. The first is to determine just the general giant sea bass density at each place. The second, I want to figure out what the prey fish density is, hoping that it remains fairly similar from site to site, so it takes out any kind of confounding competitive interactions that I might see between them. And the third is to establish to hide in if it is a rocky. Then I'm going to assess the behavior of my prey is by using baited remote underwater video cameras, or BRUVs. BRUVs have been used quite often to look at measures of fish diversity, abundance, and behaviors in many different systems. And they have been previously used to assess foraging behavior. especially in larger kind of mesopredator and predator oceanic species, especially these larger species, it's hard to sometimes observe predation events or even those facultative scavenging events in the wild. So by setting up a BRUV, it's a very standardized way to view feeding behavior for these larger animals. And another thing that's great is you can do this all without the influence of divers. So you set up your camera, you set up your bait, and then the diver goes away, and you don't have this lingering human around impacting your fish behavior. All right, so I'm gonna set up my BRUVs in a couple different scenarios of risk for my prey species. And generally in these kinds of studies, risk is measured at two different scales, or you have the option to measure at two different scales. So the first scale risk and chronic risk is defined as more of the ambient threats of predator. So the way that I like to think about this is it's kind of the background noise that a predator is providing. So it's if you go to sites where there are a lot of predators kind of lurking in the shadows, but none of them are in the immediate vicinity of a prey where they're causing any immediate danger to that prey, but they're lurking in the shadows for the prey. And the other scale of risk that is often used to measure risk for prey is the measure of acute risk, or any immediate threat that a predator is posing. So this is in scenarios where a predator is within a vicinity of a prey where it's posing an immediate threat to that prey's safety. And the way that I am going to be creating these two scales of risk, for chronic risk, I'm going to go to sites with varying natural densities of giant sea bass. And generally the way that I'm going to approach this is that I'm thinking aggregation sites are going to have high densities and therefore high chronic risk of giant sea bass, and areas that don't have aggregation sites are going to pose a lot lower chronic risk because they don't have that higher density of giant sea bass. And to pose an acute risk to my prey, I'm going to be using a 3D model predator of a giant sea bass. I'm in the process of building it right now. It's a lot of fun. Model predators have been used in these kinds of studies to pose an acute risk before. So this is a photo from a recently published paper by Emily Lester, and she used a model predator of a black-tip reef shark that looked like that. And my BRUVs are roughly going to look something like this, where I have a BRUV with 3 different horizontal baits and a BRUV that has 3 different vertical baits, so that I can ask those questions of axes that I mentioned before. So the way I'm going to do this is I'm going to go to areas with high giant sea bass density and areas with low giant sea bass density. I'm going to set up my model, and I'm going to set up my BRUV, and then I'm going to turn on the camera and watch the behavior. So I have my model set up, and then the prey at different distances from the model in areas with both high and low chronic risk. And then I'm going to do the same thing, but on the vertical axis. So I set up my predator model, and then I have the prey, or the bait for the prey, on different levels going from the model. And then I also want to do a negative control of these, where I'm just looking at kind of chronic risk at sites. So to do that, I just won't have my model, and I'll set up just my BRUV in areas with high and low giant sea bass density, and then I'll do the same thing on the vertical axis again. And in all of these trials, I'm going to leave the camera running for about 15 minutes with no divers present. I want to space them about 50 meters apart to try to avoid seeing any of the same fish on different BRUVs. And then I'm going to be looking at behaviors like time until they initially approach the bait, time until they take a first bite from the bait, and time spent feeding. So for my expected results, I'm expecting that prey in more risky scenarios are going to exhibit more risk-averse behaviors. This comes with the caveat that sometimes prey under more risky scenarios will actually start showing less risk-averse behaviors because they become more accustomed to living with a higher level of risk. So I don't know quite what that threshold is for giant sea bass, but because they're still not overly common, I'm right now operating under the loose assumption that there aren't enough giant sea bass to kind of reach that threshold of where they would be overly habituated to them as a threat. And what I think this is going to look like in terms of, exhibiting more risk-averse behaviors is in more risky scenarios, the fish are going to have a longer time until they initially approach the bait. They're going to take a longer time to take that first bite of bait because they're a little more tentative, and they're going to spend less time feeding over on the bait because they just kind of want to get their food and get out of there. And second, I'm expecting that the predicted factors that I mentioned earlier are going to influence the degree of the prey response. So if I go back to my question slide right here, we have our 7 factors. It looks like this. So for our predator factors, I think that when giant sea bass are generally in higher densities, that's going to increase the risk averse behavior. Again, kind of with that caveat that diminishing returns effect. I also think that the closer the predator is, the more risk averse behaviors a prey is going to be showing, because that's more of a danger to the prey. And I think that predators that are on a vertical axis to their prey are going to elicit a greater risk averse response than predators that are on a horizontal axis. And that's for the reason that prey on a vertical axis are a lot more vulnerable from all directions than it is on the horizontal axis. And that's been shown in studies on coral reefs. But again, there could be a caveat to this where in areas with really a lot of giant kelp, they're a lot more sheltered in the water column, so that could kind of mitigate that effect. There may be a relationship there. For my prey characteristics, I think that generally the size of the prey, the medium-sized prey, the ones that are bite-sized for giant sea bass, are going to have the most risk-averse behavior, and ones that are too small or too large for giant sea bass to eat aren't going to have as much risk-averse behavior. And I think that prey that show sort of medium amounts of sight fidelity are going to have the most risk-averse behavior. And that goes back to that idea of kind of overexposure again. So prey that are spending a lot of time at these sites with giant sea bass are getting exposed to them a good amount. But if prey are spending all of their time around the giant sea bass, then they're probably going to adjust to having them around. For my environmental factors, I think that time of day is going to matter. More specifically, I think there's going to be an increase in risk-averse behaviors around dawn and dusk. Giant sea bass, like many apex predators, are what is known as crepuscular, so they do a lot of their hunting around dawn and dusk, so I would expect that fish are a little more cautious around those times of day. And I think that habitat complexity is going to affect things. So the less complex the habitat is, the more risk-averse behaviors I would expect to see, mainly because in a complex habitat, fish have a lot more hiding places, and it's been shown that complex habitat reduce predator visibility, so that can decrease their feeding success rates. So that's generally what I'm expecting and hoping to see from this. And I think that my work has the potential to be significant because this will be, if all goes well, the first study to directly tie giant sea bass to community dynamics. So giant sea bass are not well-studied organisms because they have been so rare for so long, and what has been done on them has looked a lot at some of their genetics and a lot of their breeding, things like that, but no one's really tied them to other species yet. So that could go a long way in creating more of an argument for why it's important to conserve this species, because it makes a much stronger argument to say, yes, not only is this fish cool, but also it affects the entire system around it. So it's important to keep them around. This study is also examining the recovery of a marine apex predator in an ocean where apex predators are perpetually on the decline. That is important because you would hope that in the future, as predators continue to decline, at some point we're going to be able to start reintroducing them into certain ecosystems. So studying how that works, how that happens, how that affects the system around it is pretty important, I think. And my study has the potential to advance knowledge of non-lethal predator effects in kelp forests. So most of the research done in non-lethal predator effects has been done in terrestrial systems, and those that have been done in marine systems have mostly been done on coral reef habitats. So this is obviously happening in a kelp forest habitat, which is a little bit different, and I think it's cool. All right, so I would quickly like to acknowledge my committee members for all of their feedback, my lab for supporting this project. Obviously, the CSUN biology department is really wonderful and helpful, and my funding through the Wrigley Graduate Fellowship.